Hey besties! Welcome to Viruses and Vibes, where we talk all things viral. I'm your host, Carly Mingus, and today we're talking about one of my favorite topics, CRISPR-Cas9. So if you don't know what CRISPR-Cas9 is, it's a type of biotechnology used for gene editing. Now I know that sounds kind of freaky, but I promise it's actually really cool. After all, it's one of my favorite topics for a reason. Now I'm sure you're sitting there thinking, how does this even relate to microbiology? Well, CRISPR is all intertwined within the micro world. In fact, it actually came from microbes. That being said, let's get more into the details of what CRISPR and gene editing are. I'm going to start off by talking about some of the basics and covering basic terminology. Let's start with gene editing. Gene editing is whenever we edit an organism's DNA. I'm sure when you think of genes or DNA, you think of what makes you, well, you. And I think that's a great way to think about it. Now, our DNA kind of tells our cells what to do. It's what dictates what your cells look like and how they act, like a blueprint. To kind of give an example of what I mean here, we can talk about red blood cells. Specifically, people with sickle cell anemia. People with sickle cell anemia have abnormal shaped blood cells. Rather than being round, they're sickle shaped. This is because their DNA has a mutation which instructs their cells to build differently. Just for clarification, a mutation is a change in the DNA. For first-time listeners, you may not know what a microbe is. A microbe is a bacteria, archaea, or virus. They don't always make us sick. Some do, but some are actually beneficial. Now that those basic terms are out of the way, let's address the elephant in the room. What CRISPR stands for. CRISPR stands for Clustered Interspaced Short Palindromic Repeats. I know you're probably like, what the heck is that? What did you just say? Don't worry, you don't really need to know much past that this is describing a genome of an immune system, or what the immune system looks like. That leads me into the history of CRISPR. Where did it come from? When did we discover it? CRISPR was first discovered in the late 1900s, specifically 1987. It's actually the immune system of microbes, specifically bacteria and archaea. Now, archaea aren't as talked about near as much as bacteria and viruses, and they aren't super important for the context of this podcast. That being said, I just want you to think about them as being similar to bacteria. Now, even though we discovered CRISPR in the 1980s, no one really understood it until the early 2000s, around 2005. Then, in 2012, Jennifer Doudna and Emmanuel Carpentier created CRISPR-Cas9, a way to edit DNA. Alright, so now that we know a little bit about the history, we can start talking about what it actually does. I realize I've now said CRISPR and CRISPR-Cas9. Is there a difference? Yes. CRISPR is the system in itself, while Cas9 is the name of a protein that is attached to the CRISPR apparatus. Cas9 is often referred to as molecular scissors. CRISPR itself helps bacteria to remember a virus that infected it, so that it can fight it off if it stumbles upon it in the future, much like our own immune system. With CRISPR, though, the bacteria essentially take part of the virus's DNA and incorporate it into its own. From here, the CRISPR makes a copy of the DNA strand, which is RNA. For now, think of RNA as just a copy of a strand. This copy can bind or connect to the DNA of the virus and destroy it if it comes into contact with it. Okay, so how do these two things connect? In a genome, the CRISPR molecule locates its target region on the DNA. Then, Cas9 cuts the DNA there. CRISPR can then insert a new sequence or genetic code. It can also cut the DNA and not out of sequence, but rather cut the piece out and connect the remaining pieces. This mechanism is usually used to get rid of a bad gene. There are different ways to deliver CRISPR-Cas9 to a cell, but the one I want to talk about is using viruses to do it. These viruses can essentially carry the CRISPR system into the cell and help it get to its target. These viruses are completely safe for us to use and can't make us sick. Now for my favorite part, how it's used. CRISPR is used in many ways. Currently, it's primarily used in agriculture and medicine. I personally find the medical side of it way more interesting, so that's what I'm going to tell you about. Now, it's not widely used at the moment and is mostly still being used in clinical trials. CRISPR-Cas9 has the potential to treat many diseases that have very few or no cures. One disease it is used to treat is sickle cell disease, like I mentioned earlier. In 2019, Victoria Gray was the first person to receive treatment for sickle cell disease using CRISPR. And as of 2023, this treatment is now approved for sickle cell disease as well as beta thalassemia. Both of these are genetic blood disorders. Clinical trials are now being done to see how CRISPR can treat other diseases like cancers, blood infections, HIV and AIDS, and many other diseases. These are just a few examples, but there are so many more. Alright, y'all, I think it's time to wrap it up. I highly encourage you to go down a rabbit hole just like I did. This is truly a fascinating technology, and it brings hope to many individuals around the world. As always, feel free to ask questions and share your thoughts in the comments. References are in the description below. Until next time, I'm Carly Mingus, and I'll see you on the next episode of Viruses and Vibes.